Hey guys, so this one is going to be long. There's just no avoiding it. It is complex. It has lots of parts and pieces and pictures. It has words that you've probably never seen before, and it's something that's hard to visualize and conceptualize because we're talking about the plasma membrane, and you've never actually seen a real plasma membrane. So we're going to take it slow, and we're going to do some things in class, and I've made some models for you that hopefully will make the whole process slightly easier. But this is one of those that you're really going to have to open up that book and study, or get on YouTube and look at other videos and listen to other people explain it. So here we go. Cell communication. So all living things must be able to send messages and to communicate. It's really easy for full-grown, full-bodied organisms because we can make noises, we can shift, we can use different senses to interpret and also to send signals and, to figure out what's going on. But when you're talking about something that is as small as a cell and may be located in one region of the body and needs another cell on the opposite side of your body to do something, it's kind of an issue, especially when you don't have a mouth. So hence why cells use what we call a signal transduction pathway. Think of it as a very complex note-passing system. Think of when you were little and you wrote notes in class. You would give it to so-and-so, to give it to so-and-so, to give it to so-and-so, to give it to so-and-so, and to give it to so-and-so before Susan could get the note that you wanted her to see. And then she would write something in response and pass it all the way around again until it got back to you. That's kind of how cell signaling can work. There are some different ways. Some are very simple and some are highly complex. So a signal transduction pathway is what a cell is going to use to send and convert signals. And it's going to happen starting at the surface of the cell, so on the cell's actual plasma membrane. That signal is going to start off by interacting with a protein on the cell surface. That protein is going to release or elicit a response that's going to take place cellularly, so inside of the cell. And that message is going to be passed on and passed on and passed on, mostly by proteins and enzymes, until the cell responds in the appropriate way. Now, when you consider organisms that are prokaryotic and eukaryotic, that are multicellular and unicellular, and you compare our most common ancestor, which by the way was alive about 1 billion years ago, you'd realize that our signal transduction pathways are not that different. So the pathways that prokaryotes use are pretty much the same pathways that eukaryotes use, which suggests that this process is so well evolved that we didn't have to make very many changes. Even in this modern day, it's still the same way that our cells talk to each other. So let's start off with something simple. Yeast cells use signaling for one main thing, and that would be to correspond sexual reproduction. So in yeast cells, you have two mating types. We have A cells and we have alpha cells. Both of them make signal molecules called factors. So an alpha cell makes an alpha factor, but it has a receptor for an A factor, whereas an A cell makes an A factor, but it has a reception site for an alpha factor. So what happens is when the two cells are ready to mate, they will start making their A factors and their alpha factors, and they will release them through their plasma membrane out into the environment in which the yeast exists. Once those alpha factors come into contact with a receptor on an A cell, it suggests to that A yeast cell in which direction a potential mate is located. So the yeast starts growing in that general vicinity. The same thing is going to happen to the alpha cell. When an A factor comes into contact with its cell receptor or its protein receptor, it is going to suggest that a possible mate is in a certain direction, and the alpha cell will start growing in the same vicinity that it got the factor from, until eventually the two cells meet and they will go through meiosis and they will mate. All right, so this exchange just keeps going until reception happens, so until the factors bind to their respective to their respective receptor sites, and then it will cause the two cells to fuse to each other, thereby syncing their genetic information, which are different from each other because one's an A cell and one's an alpha cell, and then meiosis is going to occur. Another type of very simple cell communication is a process called quorum sensing. Quorum sensing tends to be used mostly by bacteria and prokaryotes, and it is all about determining population density. How many individuals are around me, and if so, how should I respond? Should I keep reproducing? Should I keep doing binary fission? Or should I stop reproducing? So here's how it works. Cells are going to, so these are bacterial cells. Hold on. See if I can get this drawn. Bacterial cells are going to emit a signal molecule. Let's just make them blue and circular. Okay. Those signal molecules have a specific receptor on the cell membrane of other bacterial cells. When they connect, it's going to send off a signal internally, and that cell will know, based on how many signal molecules it received and how long it took those signal molecules to get there, whether or not there are a lot of bacteria around it or a little bit of bacteria around it. So if there's a small amount, they're going to keep dividing through binary fission until they evenly cover whatever container or whatever environment they're in. Now, if the message gets there quickly and there is a lot of the signal molecule, it triggers the shutdown of reproduction in the existing bacterial cells. So they stop producing individuals and they stop overpopulating the area. Quorum sensing especially is related to food sources. So if the population is too big, then the food source is going to be quickly utilized and the population will eventually die or will be damaged as a result. So density numbers are going to be dependent on how much available food and other resources, or how available, food and other resources are. So when we have cells in a multicellular organism, these cells are going to have to communicate via chemical messengers. And there are two ways that it can happen. We have what we call local signaling, which is kind of like calling locally, or we have long-distance signaling, which is kind of like calling long distance, so like calling Dallas versus calling New York. For animal cells and for plant cells, they have a special feature called cell junctions that allow the cytoplasm of cell A to be in direct contact with the cytoplasm of cell B. That way, any messengers that they're making can literally just diffuse from cell B into cell A or from cell A into cell B. In an animal cell, that feature is called a gap junction. In a plant cell, that feature is called a plasmodesmata. But again, it's just a direct connection of the cytoplasm between two adjacent cells. Another way that we can have local signaling is through cell-to-cell recognition. So all the cells have receptors on their membrane. Some receptors can fit together, like it shows in this picture. When that happens, it may trigger a response in the target cell. So that would be another example of direct contact or cell-to-cell recognition. Both of these are local signaling. We're not moving very far. We're right there next to each other, just like if I called within Mesquite or within Dallas. Now, in other cases, animal cells especially might communicate using what we call local regulators. So these are messenger molecules that will travel, but they'll only travel a short distance. So going from Mesquite to Garland, for example, that would be a short distance versus going from Mesquite to Wichita Falls. That wouldn't be a short distance. So examples of local regulators or parts of your body that will do local regulating would be some of our glands, like our paracrine glands. They will release messengers that when they get into contact with their target cell, it's going to have some effect. It's going to cause that cell to do something or to differentiate or to change its shape or whatever. Another example would be synaptic signaling, which we see in our neurons. And when we talk about the nervous system, we'll talk about this in a whole lot more depth. But this is pretty much the passing on or the propagation of an electrical signal. The problem is when we get to this section of our neuron, we have a space. And the electricity can't jump through the space. The electrical signal cannot jump this gap. So instead, we make a neurotransmitter that takes the same signal and passes it on to the new neuron. Examples of long-distance signaling will happen in both plants and animals, and they will utilize chemicals called hormones. So literally, one cell will make a hormone. The hormone will get into the bloodstream, so it's kind of like the body's version of a highway. It will travel until it gets to the target cell. It will send its message off and result in some kind of change. A good example of this would be the maintenance of like our blood sugar level. So let's talk about what these hormones can look like. And specifically, we'll talk about animal hormones. So animal hormones will come in three forms. There are steroid hormones like testosterone and estrogen. And since they're steroid, that means they are lipid-based. Then we have modified amino acids like thyroxine, for example, which is a hormone that your thyroid gland makes, but it comes from an amino acid called tyrosine. So it's a modified amino acid, so it's protein in nature. And then we also have proteins that will make hormones, for example, insulin. So those are the three main kinds of animal hormones, sorry. So now let's talk about how the signaling actually works. And we know what the signaling is. We know general information about how it works, and we know why your cells do it. Let's talk about how it happens. So there are three stages or three steps to cell signaling. The first step is reception, the second is transduction, and the third is response. A lot of these cell signals are going to start exterior, so on the outside of the actual cell. The initial place of contact normally happens on the plasma membrane or the cell membrane. So again, reception, transduction, response. Let's step through it. Starting with reception. So in reception, we're going to have a signal molecule, also known as a ligand, bind to a receptor. Now, signal molecules or ligands can be hormones, they can be neurotransmitters, or they can be factors. The receptors are very specific. They're not going to bind just anything that happens to float by and end up in their little active site. They're going to bind specifically to certain signal molecules. Once that molecule binds, it's going to result in a conformational change of the receptor. So the receptor might actually move, or it might change the shape of its active site or the whole protein, and that is going to start off the transduction of our signal. All right, so that's step one. Now, most, if not all, of your signal receptors are going to be plasma membrane proteins. There are some that are internal, but that's a little bit of a different situation, which we'll talk about later. All right, so let's look at these receptors in a little bit more depth. There are three main types of membrane receptors. They are G protein coupled receptors, receptor tyrosine kinases, and ion channel receptors. We're going to specifically talk about G protein receptors and ion channel receptors. Okay, so a G protein coupled receptor is going to be found on your plasma membrane, so it's a plasma membrane receptor, and it works with the help of a G protein. So it's just a type of protein called a G protein. The G protein is going to be the molecule that's actually starting your signal. So it behaves like an on or off switch. Now I know you're used to thinking of ATP as the only molecule for energy that your cell has. But remember when we talked about modified amino acids, we said that technically any nitrogen base can become an energy molecule. So G protein is activated by the presence of one of those modified amino acids called GTP. So GTP just stands for guanine triphosphate, just like ATP is adenosine triphosphate. So GTP is going to be an energy molecule. Just like how ATP can lose a phosphate and become ADP, GTP can also lose a phosphate and become GDP. So here's the rule. If the G protein has GDP bound to it, the protein is inactive and it's not going to do anything. It gets activated by a phosphate group attaching itself to GDP and making it GTP. That's going to give the protein the energy that it needs to start off the signal. All right, so this is kind of what it looks like. Here is our receptor. This is our G protein receptor. This is our G protein. Because it has GDP bound, it is inactive, so that means it's not actually working. So our signal molecule is going to come in. It is going to activate our receptor, which is going to trigger the formation of GTP for energy. Once GTP has been made, our G protein is going to slide along the plasma membrane until it comes into contact with an enzyme. Now note, while GTP is being made, the enzyme isn't activated. It's in its inactive form. Once our GTP coupled G protein meets the enzyme, it is going to activate the enzyme, and that enzyme is going to start a cellular response. Once the response has started, GTP is going to lose its phosphate, it's going to become GDP again, and everything goes back to the inactive state. We have a model that looks exactly like this with parts that actually kind of move around that we will utilize to help you understand this even better in class. All right, the other kind of plasma protein receptor that I enjoy talking about are ligand-gated channels. So a ligand, like you know, is just a signal molecule. So what it is, the ligand-gated channel is a protein pore that's found in your plasma membrane, and it exists in two formations, either open or closed. It is going to either open or close in response to some kind of signal molecule, so your ligand. So if you look at the picture, it's much easier to understand with the picture. So the problem is we have these ions that need to get into the cell. The ions are charged, so they're not going to easily be able to pass through this cell membrane. They're going to have to go through a special receptor, which is what this ligand-gated channel is. But right now, the channel is closed. The pathway is blocked by this section of the protein. When the ligand comes in and binds, it causes the channel's gate to open up. So the little part that was closing off the channel will lift itself up and open up that protein. Now the ions can flow through, and once the ions are inside, they're going to start off whatever the cellular response is supposed to be. When enough have gone through, the ligand is going to unbind and leave the protein, and the channel goes back to that closed position. Now, these ligand-gated channels can exist on plasma membranes, so they're leading into the actual cell or out of the cell, but they can also exist on the plasma membranes of organelles. Okay, so we've started off the process. Our signal has bound itself to the receptor. We have some kind of initial or initiation taking place. Now what happens? Well, like I told you, not all of the receptors are going to be on the surface of the plasma membrane. Some of them are intracellular, so they're found inside of the cytosol or even in the nucleus of the cell that we want the response for or from. That cell is called a target cell. These messengers tend to be small or hydrophobic, which means that they can easily move across that membrane and activate receptors. So that's why their receptors don't need to be on the outside, because the messengers can get into the cell. They tend to be steroid or thyroid hormones in animals. Once they are inside, they're going to activate a receptor, and they're going to form what is called a hormone receptor complex. So the hormone binds the receptor, and then the two of them are going to become a transcription factor, which will turn on specific genes to be made into a protein. And that would be intracellular receptors or steroid-based receptors. All right, so this is what it looks like. So here's our hormone, which is on the outside of the cell. Because it is hydrophobic and it is small, it can fit through the plasma membrane and find the receptor protein that it needs to find and activate. Once it binds to the receptor protein, we have now formed an activated hormone receptor complex. This particular hormone receptor complex is going to become a transcription factor. It's going to activate certain genes. Well, our genes are found in our DNA is found in our nucleus, so it means that the complex needs to go into the nucleus through a pore and sit on a section of DNA, thereby activating that section of DNA for transcription. Once transcription happens, we will form a piece of messenger RNA, and then through translation, after that messenger RNA has left the nucleus, we will form the new protein, and that will be the response that this hormone was trying to get. So, transduction. What's the whole passing on of the signal about? Transduction is normally going to be a multi-stepped pathway. It's multi-stepped because that allows the signal to be amplified. It makes the signal bigger. All right, so we get a bigger response. Excuse me, a more efficient response. Along with making the response bigger, multi-stepped pathways allow lots of opportunities for the coordination and the regulation of responses. So that way, several cells can all give the response needed, like in the case of us trying to lower our blood sugar, and we want to release lots of insulin to take care of that situation. If one cell released insulin, that wouldn't be enough to lower our blood sugar level back to what normal is. At the same time, if the pathway is going and we're getting the response we need, but now we need to stop the response, we need to regulate the response, there are many steps along the way that we can stop it, tone it down, or regulate said response. Okay, so of course it's going to happen with the help of proteins. Proteins are going to relay the signal from the receptor, where the signal was interpreted, to the actual point or part of the cell that is responsible for making the response needed. The receptor activates a protein. After that little signal molecule lands on it, that receptor is going to activate a protein. That protein will activate another protein. That new protein, another, and another, and another, and another, and another, and another, until we end up with the desired outcome that we are seeking. At each step, the signal is transduced into a different form. So this normally means that we're making energy or we're using energy because we're going to be undergoing some conformational changes of some sort within these proteins. So just to recap, once the signal molecule lands on the receptor is going to activate a protein, which activates a protein, which activates a protein, which activates a protein, which activates a protein, until we get the response we need. It's just like when you were little and you were passing notes. You wrote your note. You passed your note to John, who passed it to Susan, who passed it to Lucy, who passed it to Frederick, who passed it to Tina before Jerry could get it at the front of the room. Same thing with your signal transduction pathway. So, how exactly does it work? Well, the signal gets transmitted by one of two processes, either phosphorylation or dephosphorylation, and both of these are going to happen on protein. So we call them protein phosphorylation or protein dephosphorylation. There are two enzymes which are important because they are in charge of either phosphorylation or dephosphorylation. So let's start off by talking about the names of the enzymes, and then we'll talk about what those two processes are. Phosphorylation is controlled by an enzyme called a protein kinase. Okay, so again, a protein kinase is an enzyme that is in charge of or coordinates phosphorylation. What phosphorylation is, it's the transfer of a phosphate group from ATP to a protein. So ATP loses a phosphate group. It's becoming ADP. Dephosphorylation is controlled by the protein phosphatase. And what protein phosphatases do is they take phosphates from proteins. So that's dephosphorylation. So let's do it again. Phosphorylation is done by protein kinases. We take phosphates from ATP and we give them to proteins. Dephosphorylation is done by protein phosphatases, and they take phosphate groups from proteins, and normally they give it back to ADP, turning it into ATP. So it's almost like a switch, on, off, on, off, on, off. Phosphorylation is on, dephosphorylation is off. Phosphorylation is on, dephosphorylation is off. So it helps to activate the signal. Either the protein's activated so we can keep sending that signal on, or the protein is deactivated and the signal stops moving. We don't send it on anymore. So kind of like the teacher noticing that you are passing a note. If the teacher notices and she goes and stands between John and Freddie, then John can't pass a note to Freddie. Your signal has stopped. But if the teacher doesn't notice, and normally because she takes the note away, if the teacher doesn't notice, then the note keeps being passed. All right, so this is a visual to go with it. So here's our signal molecule. We've landed on our receptor site. It's been activated, and we activate a protein. A protein kinase is now going to become activated. It is going to take a phosphate group away from ATP and activate another protein. And then it's going to get dephosphorylated. So we're phosphorylating, dephosphorylating, phosphorylating, dephosphorylating, phosphorylating, dephosphorylating. And we're just going to keep going until we get to that final protein that is going to give us the response that we are looking for. Okay, so what is the response? Well, the cell's response is what it does as a result of the signal that it is getting. It is the output that that cell is going to give. What normally happens is this transduction pathway is going to allow your body to regulate one or more of its cellular activities, like transcription or translation or recycling used up materials so that they can, or old materials, that they can be used again, making something new, sometimes not making something new. It just depends on what your body needs. The response can happen in two places. Either it's going to happen in the cytoplasm, or it's going to happen in the nucleus. In a lot of our signal pathways, we are going to be regulating either the making of enzymes or other proteins, and this is done by either turning genes on or turning genes off. We turn the genes on, we can now make that new enzyme or protein. We turn the genes off, we stop making that enzyme or that protein. And overall, especially for those hormones that are steroid in nature, they're going to result in a molecule that is going to be a transcription factor, something that is going to get a section of DNA to be made into a functioning protein. right? So visual to go with it. Signal binds, receptor gets activated, we activate a protein through a series of phosphorylations and dephosphorylations. That message gets passed on until we end up activating a section of gene that's going to become a piece of messenger RNA that is going to become a protein that will do some kind of function. Now, while some pathways are going to regulate the synthesis of enzymes, other pathways are going to regulate the activity of enzymes that already exist. Okay, so they're going to stop the enzyme from doing a particular job or function, or they're going to allow an existing enzyme to do a particular job or function. Signal pathways can also affect the physical characteristics of a cell. For example, the shape that that cell may take on. All right, so how do we tweak the response and make sure that it gets exactly where it needs to be and it's as big as we need it to be so that we can get exactly the type of response that we want? Well, multi-step pathways have two main benefits, which is why your body does it. Benefit number one is the signal is amplified. You have lots of molecules carrying the signal, which means you're going to have a lot of response to those molecules. The second reason that it's important, or the second benefit to having a multi-step pathway, is that we can contribute to, whatever, the American pronunciation, whatever, to the specificity of the response, making sure that that cell responds in a way that in a way that the situation warrants for. We don't want just any kind of crazy response, any kind of crazy activity. We want a response for the current situation, for what we're trying to get. Enzyme cascades amplify cells' responses. So when we talk about the response being bigger because we're getting more and more molecules involved, that's exactly what an enzyme cascade does. At each step along the process, what we activate is going to be greater in number than what we activated in the step before. So one signal molecule might come in and activate one receptor. One receptor might activate one protein. That one protein might activate two proteins. Those two proteins might activate four proteins, and it just keeps building and building and building until we end up with the type of response or the kind of response that we want. Now, there's a special kind of protein that really helps with the signal amplification, and they're called scaffolding proteins. They're really big, and they're pretty much just relay centers. So this would be your scaffolding protein right here, the pink one. And on that scaffolding protein is going to sit three smaller enzymes or three smaller proteins. Once this scaffolding protein gets activated, it is going to activate all three of these, which are going to leave and activate more and more and more and more and more and more. So scaffolding proteins increase the efficiency of how the signal is transducted by grouping together different proteins involved in the same process. So instead of having to go and find protein A, and then find protein B, and then find protein C to get them all activated, you activate the scaffolding protein. It activates A, B, and C all at the same time. Okay, so different kinds of cells are going to have different collections of proteins. Mostly those proteins are going to be of one of two natures. Either they're stimulatory, so they're going to increase response, or they're inhibitory, they're going to stop response. Because of the presence of all of these different types of proteins, it's going to allow individual cells to detect and respond to different signals. Because don't think that this is one big coordinated effort in the sense that, okay, only the insulin signals are going to go out now. All right, now only the growth factor signals are going to go out. No, signals, all kinds of random signals are always being sent to different to different cells. So the cells have to be able to interpret and detect what those different signals want them to do. Even when you send the same signal, it can have different effects on the same cell depending on which protein gets activated and which pathway is utilized. Proteins and cells have this thing called pathway branching, or what we call cross-talking, which help the cells to coordinate the incoming signals. And I'll show you what that means in just a second. All right, so here's an example of what I mean by cross branching or talking. This is the normal pathway. Signal molecule comes in through a series of activations. We activate lots of proteins, we get the response that we want. So this pathway leads to just one type of response. At the same time, we can have the same signal come in. It will activate a protein, and that protein can activate two different types of proteins, which will then go through their process, and we will end up with two different responses. So this would be an example of how the pathway can branch and lead to, instead of one response, we have two responses. Another example, this would be an example of crosstalk, is where two different signal molecules activate two different receptors and start two different pathways. But one pathway actually is going to regulate the other pathway, either by activating it or inhibiting it, resulting in a response. Then the last way is when we have a different kind of receptor being utilized by a signal molecule. So the signal molecule comes in, it lands on a different receptor to the one that it would normally land on. It activates a totally different pathway. It ends up with a response of some kind. So we know how the signal starts, we know how the signal progresses. How does the signal end or terminate? Inactivation is pretty much the last step of the whole signal pathway. So when molecules leave the receptor, when that ligand or that signal molecule or that hormone is no longer bound to the initial receptor, the receptor goes back to its inactive state. And when that happens, the transduction stops. That's it. It's the opposite of the initiation step. Apoptosis is a great way to study cell signaling and cell communication. Apoptosis is programmed or controlled cell suicide. Pretty much it's programmed cell death. And in the process, a cell is pretty much sectioned off and broken up into pieces, packaged into vesicles that have digestive or hydrolytic enzymes in them, and all the parts are digested and recycled and reused. Apoptosis prevents enzymes from leaking out of a dying cell and damaging the cells that are healthy next to that dying cell. A good example is the interdigital tissue that we all have between our fingers. When you were in the womb, your fingers looked a lot more like fins than they did actual fingers. And you know that you can still see the webbing or the tissue that link your fingers at the base. Well, imagine that webbing all the way up at the top, the very, very tips of your fingers. All of that tissue had to be killed off or died off somehow. It happened through apoptosis. Normal cells all cells as a matter of fact, have a receptor for a death signaling molecule. Now, if the cell is healthy and normal, doesn't need to be killed for whatever reason, it is going to have a protein in its mitochondria called CED9, which when active is going to inhibit the activity of a different protein called CED4 and another one called CED3. When a cell needs to undergo apoptosis, a death signaling molecule is going to come and bind to the receptor. Once that binding takes place, a signal pathway is going to occur, which is going to inactivate CED9. When CED9 inactivates, a couple of things happen. CED4 and CED3 are going to then become active, but this is how it happens. CED4 becomes active first. it is going to start a signal pathway that is going to activate CED3. When CED3 is activated through a series of cascades and amplifications, it is going to activate two types of enzymes, proteases and nucleases, both of which are digestive enzymes. They're going to start breaking apart that cell. All right, so capsases are the main types of proteases. Proteases are enzymes that cut up proteins, and these are the enzymes that are going to carry out apoptosis. Apoptosis can be triggered by, like I showed you in that picture, some kind of death signaling ligand, or we could have severe DNA damage in the nucleus, or we could have protein misfolding in the endoplasmic reticulum. Any of these three things will trigger apoptosis. Now, apoptosis is one of those processes that we think developed really early in animal evolution, and it is highly essential for the development and the maintenance of all animals. But in recent times, we've found that it's linked to some certain types of diseases, especially some of the chronic ones that we really don't have a cure for. So, for example, Parkinson's and Alzheimer's. Apoptosis interference can also contribute to some kinds of cancers. Cells naturally know when they've reached their dying point, and sometimes they will just naturally trigger apoptosis in themselves. A cancerous cell, however, will never do that, thus it continues to grow out of control. Cell communication allows for the coordination of individual cell processes. Sometimes these processes are going to support functions of the entire organism, like when your body is deciding between I need all of my available energy or I have extra energy that I can store. So available energy is going to be in the form of glucose that we will utilize, but if we have excess, we can store it in the form of glycogen. And your body will hold on to it until such a time where you don't have glucose to be utilized, and then it's going to convert from glycogen back into glucose. This whole process happens through cell communication. Something else that can happen is your cell communication can trigger either internal or external cues, like your fight or flight response. Either you get really scared and you run away, or you get filled with adrenaline and you can do things that you normally or ordinarily wouldn't be able to do by yourself. So let's look at the action of one type of ligand or signal molecule called epinephrine, also known as adrenaline. So we're going to try and take it through slowly. So we're looking at how we go from glycogen to glucose in this as well. Because if you think about it, adrenaline keeps you pumping. It's kind of like that fight response. You know, you can lift the car off your mom if she's dying or whatever. You're going to need some energy to be lifting that car. So one of the things we're going to have to make is some glucose. So the signal is epinephrine, also known as adrenaline. That is your signal molecule. Now, this is going to be non-steroid in nature because notice it is binding to a receptor on the plasma membrane. It's not going through the plasma membrane, but it's binding on the outside of the cell. So epinephrine is going to come in. It's going to bind to its receptor, and it is going to activate that receptor, which in turn is going to activate GTP, the energy molecule. GTP is going to activate G protein, and G protein is then going to activate an enzyme. This enzyme is called adenylate cyclase. Adenylate cyclase's job is to activate CAMP, which is another type of signal molecule. CAMP is made from ATP, by the way. So, through a series of phosphorylation and dephosphorylation, so here we're phosphorylating, here we're dephosphorylating, here we're phosphorylating, we're going to finally activate an enzyme called glycogen phosphorylase. Glycogen phosphorylase, so this is our transduction, Glycogen phosphorylase is going to take glycogen and convert it into usable glucose, which is the response that your body wants. Once the glucose is made, it's going to be released into your blood. You now have the energy to lift cars and jump mountains and do whatever else it is you need to do. All right. So another one we could look at would be the regulation of blood sugar. And we know this is going to happen using insulin. So insulin is going to be our signal molecule or our ligand in this situation. It is produced by islet islets of Langerhans cells or beta islet cells, same thing. Insulin is going to be released whenever your blood sugar level gets above 90 milligrams per 100 milliliters. Above 90 is considered high. So when your blood sugar level spikes, it gets high, it is going to cause these Langerhans cells to release insulin. Insulin is going to circulate throughout your bloodstream, and it's going to cause a couple of things to happen. Your body cells are going to start taking up or utilizing the sugar in your blood. If there is excess sugar, your liver is going to store that in the form of glycogen, and it's going to send a signal to your brain telling you you are no longer hungry, so please stop eating. This is going to happen until your blood sugar level gets back within normal range, but if it keeps going on, then the opposite will happen. Your blood sugar level is going to fall. It's going to go way below 90, which is considered low blood sugar levels. So now another response is going to take place. This time, the Langerhans cells are going to produce a different type of hormone. This one is called glucagon. Glucagon is going to cause your liver to release some glucose, which will elevate your blood sugar level. And remember, we're trying to get it within this range. And it's also going to trigger your brain and tell your brain, hey, you're hungry, you need to take some food in, which will also get some glucose back into your system. So your body is constantly going between this extreme and this extreme. We're trying to keep things within normal. But none of this would be possible without cell communication and those transduction pathways. All right, and we're done. So when you get to class, we're going to look at it some more. We're going to watch some videos. We're going to play with some models, and we'll talk it through if we need to. It's one of those things, like I said, that's kind of hard to comprehend the very first time you see it. You guys really are going to have to read your book and look at other videos and get the UT homework that I'm going to schedule and tell you about tomorrow done. I hope you have a great night. See you tomorrow.